Thanks. It's quite an honor to be here and to follow behind some of the speakers that we just heard. This is, okay, Chris, you make me feel like a duck out of water. So I'm an animal scientist, and I'm going to share some of the data that we've worked with just recently, and it does relate now to some of the work that we've done with FGF23, a little bit of background as to why we got into that, but would like to make one comment if I can really quick first. It's really, over my career, I found out that something that really brings an honor is when you see students that you work with excel. And I just acknowledge Chris and what she's worked with, had the opportunity to work with her during her PhD program and some since then. And it's really great to see what you've done with this society and the people that you're working with here. So it's good. I really appreciate it. Thanks. So some of the work that we've done, I'm going to try to move through this as fast as we can, see if I can help a little bit with some of the computer glitches that we had, and I don't want to get into one of those. If I can make the arrow button go on this. I want to first acknowledge, oh, that's really sensitive. It is going to make me move along fast. On the slide with the authors were 3 students that I've worked with, and they've really contributed to the work that we've shown. So just wanted to acknowledge them. Mariola, most of the work that we've done is a postdoc work that she's done. She's now at Indiana University Medical School, continuing to work in the area of FGF23 and phosphorus regulation. Laura and Brittany. Brittany's a current graduate student with me. Laura's still a scientist now that's working in the lab with me. So, I'm not hitting the right button. Green. There we go. So it looks like I'm going to cover a lot. I'm not. Okay. What I really want to focus on, and I'm an animal science, a little bit different background, different perspective that I come to, and I'm going to try to point out some of that perspective, the difference between animal nutrition and what you might do in human nutrition and how we think about things. I want to make those points just briefly, but most of what I want to focus on is this area of dietary phosphorus as a target regulator of vitamin D, phosphorus, and the FGF23, that whole axis. And there's some data that I think you might be quite interested in, especially with the previous presentation by Dr. Cole. Hope to be able to stimulate your thoughts a bit and challenge, maybe not to try to bring you into my world, but to take some of what we might experience with this and bring it into your world. Let me just mention with phosphorus some of the background and differences. In animal agriculture, we're concerned about phosphorus and environmental pollution. And so we have a need, and I'll just make this point now, in animal nutrition, phosphorus is typically limited. In calcium, we have plenty of calcium that we can put in the diets. We control the diets of the animals. So calcium is usually not a limiting feature in the diet that we feed to our animals. So concept now that we're different in, as I've challenged my colleagues in the animal agriculture area, we mostly focus on extrinsic factors. Those are factors that would be involved outside of the animal, the diets that we feed, and as I just mentioned, in our world, phosphorus is often a limiting feature. The availability of the phosphorus is a big issue, and we have approached that now from the standpoint of trying to improve the digestibility, the availability, trying to decrease the excess phosphorus that's excreted by the animal that ends up in the lakes and streams and causes surface water pollution. So there's a lot of pressure involved in that world. Calcium is usually in excess. We can very inexpensively add calcium to the diet. We control the diet. So calcium is not a limiting feature typically in the diet. The ratio of calcium and phosphorus, yes, that can create problems. But what I think we've ignored when we try to only determine the digestibility of phosphorus are some of the intrinsic pathways, what happens within the body. And this is just a simple diagram now to look at phosphorus homeostasis that would be there. We can often try to measure that, but I'm not sure it does much good because you know the flux then of phosphorus in and out of those pools through the kidneys that this group now would certainly be talking about. My role and the thing that I tend to think about most is actually the bone area. Certainly tissue accretion, we get into production animals, milk excretion, growth and tissue accretion, that would certainly affect the use of phosphorus. And you certainly know the role of the kidneys in regulation of that, but we don't think often enough about what happens in the bone. And what Dr. Cole just mentioned, that what's going on with the bone, some data now with the FGF23 story where we're showing changes in the CYP regulations of activation and degradation of vitamin D at the bone level. Real quick, now part of what got us into this, a little bit of background. I get concerned we're in a dairy state, so hypocalcemia that occurs in dairy cows. And what you're trying to do is you actually decrease the calcium in the diet in the dry period before the calf. And I get a lot of questions, do we have that problem with sows? There's a major problem that goes on in the sow production world with mortalities and what is often perceived by the veterinarians as a hypocalcemic response. So we set up an experiment just to challenge that, to question that, and can we alter calcium response, calcium homeostasis right around parturition. So this is gestation day on the bottom axis there, day 113, about 2 days before parturition, lactation day 1, 3, and 5. We measured with catheterized sows, then at 7, 10, 1, and 5 o'clock, each one of those time points we measured serum calcium at 15-minute intervals. So each one of the dots has 15-minute intervals average within that. Knowing what we know from the dairy cow, and overfeeding calcium, we try to set up a situation where we can actually stimulate the hypocalcemic response. Within the physiological range, the dashed lines with either total calcium or ionized calcium circulation, there's no evidence of a hypocalcemic response in this critical time period. So the animal is very readily able to maintain serum concentrations of calcium. Phosphorus, which raised the question. So with high calcium that's in the diet, and this would be the triangles that we're showing on here, the animal's not able to maintain with excess calcium on the diet, they're not able to maintain the serum phosphorus concentration. That raised a lot of questions, stimulated some thoughts in our mind, along with other things that we're working on. Okay, and this is a picture of our humpback pig, or a pig with kyphosis. Starting in 2004, 2007, and our research herd is a very controlled unit. 2007, we had about 30% of the herd with no changes in genetics or health status of the animals. 30% of our animals ended up with a humpback condition. We've worked on that since that time period and continue to work on that. We have orthopedic surgeons that are working with us that are using this as a surgical model to correct kyphosis. So the bone is the focus, other lesions that we see, that's been a major focus in our lab since that time. But at the same time, how this phosphorus regulation, what that led me to, with I teach comparative nutrition class to our majors. And in 2008, I would have claimed that I really have a great understanding of vitamin D, calcium, phosphorus. My knowledge of that, my understanding is 100%. So these humpback pigs has changed, and we have a diminishing returns, diminishing knowledge that's occurred in my sense. And one of those, if you think about the time frame of what we've known about FGF23 and its role in phosphorus, 2005 was kind of an introduction to that. This is 2008 at the Bone and Mineral Research Society meetings. I heard this new term, FGF23, and I didn't even know how to spell FGF23. Okay. Did not know what it was. And we began to dive into that a little bit more, trying to understand, and there's something that's going on there that we could not explain. And the more I got into it, the more complicated it got, and all kinds of different things that I didn't really quite understand about how calcium, phosphorus, and vitamin D would be regulated within the body. So that had to force me to kind of redraw the diagram of homeostatic mechanism and recognize the FGF23 is actually produced in the bone, targets the kidney, and you, as an audience, know this, so I'm not going to spend a lot of time going through what that does with the kidney, and it's just talked about in the previous presentation, where it is involved now with the translocation of the sodium phosphate transporters, but also the activation and degradation in controlling vitamin D homeostasis. So that humpback pig model, if I go back to that just for a moment with your thought, we actually know now that that was due in a maternal diet to the depletion or the failure to add vitamin D into our diets. So we have substantial evidence that the kyphosis condition is caused by maternal vitamin D deficiency in the herd, and also slight changes that we can make in the nursery pig, the neonatal pig, with calcium increasing the phosphorus in keeping the vitamin D out. So that's been an area that we've looked at, and we set up an experiment because some of our attempts to measure serum concentrations of FGF23 have not been successful in the pig. We measured in humans, we can measure that in mouse models that are being used. We tried all kinds of different approaches, and we're not able to measure, even though there's not 94% homology between the porcine FGF23 sequence and human and mouse sequences, very similar. We tried the mouse kits on pigs, and we're not successful. I think I have some ideas, but this experiment was actually designed in part to address that because of the time course that these changes are occurring in the whole animal. So we set up an experiment. We fed a low phosphorus diet for 4 days, switched from a controlled diet to a standard diet, and then immediately after that, looked at 3, 6, 12, 26, 36 hours after, looking at blood, urine, and tissue collections of pigs over that time period, and then out to 5 days. So this is the time frame of the experiment and how rapid these responses are occurring within the pig. If we look at plasma phosphorus, we've got on these graphs then the plasma phosphorus and urine phosphorus concentrations. We're interested in showing, does that shift, which occurs first? Are we seeing changes in plasma concentrations? Are they preceded in the first graph? We kind of infer that changes in the plasma concentration with that tight time course actually precedes then what we see with urinary excretion. With plasma calcium, not as much change there, but still a tight time course. We've got the plasma PTH values, and those are going up, and returning then to baseline within the 5-day time period. That has to do in part with our challenge to try to measure then the serum circulating concentrations of FGF23. Some of our attempts earlier, we would wait 2 weeks after an adjustment to the diet trying to make the measurement, and have we already lost the response? So we measured both the mRNA in the kidney and cortical bone now, looking at the FGF23 response. Sorry, one of the bars, 2 of the bars seem to have lost out here, but there's 0, 12, 36, and 108 hours after the switch of dietary phosphorus. The panel on the right of the screen there shows the control animals at 1, and then a dramatic increase in the mRNA expression in the kidney and in the bone over the time course. Several experiments we've measured the mRNA for FGF23 in the pig bone. We can certainly detect changes in magnitude-fold increases in the FGF23 mRNA. And if you look now, we measured the sodium phosphate 2A transporters in the kidney and the brush border, isolated the brush border membrane, and looked now within that membrane, so it's a translocation of those proteins in the kidney. The control animals, so with increase in the 12, 36, and 108 hours, we would show that the transporters have remained then translocated then within the kidney brush border membrane. What is of interest now for our world in trying to track this and tie this back into even the kyphosis response within the pig is the change that we're seeing in the kidney activation and degradation of vitamin D. So we look at the CYP27B1 for activation, degradation of the CYP24s, and so we're looking at the mRNA expression in the kidney and in the bone tissue. And this data was a little bit surprising at first. We began to look at the raw results in the lab, and the first acute Mariola, who was a postdoc now that did this, that did she mix the samples up? Did we have the wrong sample? Did we make sure that we had the labels, the samples labeled correct? But what happened now within the activation of vitamin D in the far panel there, the mRNA in the kidney actually immediately went up within the 12-hour time period, and then by 108 hours, 5 days, the activation of vitamin D had gone back down, and so as the animals adjusted to that insult or massive dose of phosphate, what's occurring within the kidney. Degradation of the vitamin D actually increased, so the CYP24 that would be involved with degrading the 25-hydroxy to the 24-25-hydroxy, that degradation increased in the kidney. The opposite response, even though the magnitude is not quite the same within this, the activation in the bone, and this is what was confusing. The bone is saying, well, activation of vitamin D is increasing over this time point where the kidney is largely decreasing. And then I begin to think about the model and homeostasis and the attempts of what the body's doing to try to regulate the plasma concentrations. And it began to make sense that the kidney's trying to get rid of the excess phosphorus. The bone actually can help with doing that by actually sequestering and pulling more, depositing more phosphorus within the bone material. So a change in the flux of that we're seeing through the tissue is our explanation that we're trying to use with it. Degradation of the vitamin D actually then a decrease at the bone level. So the body's not degrading at the bone, and it's an extrarenal response, and how much the kidneys involved with regulation of vitamin D versus extrarenal tissue. Certainly the bone is playing a role in this particular case in responding to that. So further evidence to support that is actually shown with the serum concentration now of the 25-hydroxy. That's the level that we measure to indicate adequacy of the status within the body. In the 25-hydroxy, there were no changes over this time course, over the 5-day time course, in circulating levels. And that might not be too surprising given the half-life of 25-hydroxy in circulation. But we did see differences now with the 24-25 that's consistent now with what we're seeing with the changes in the mRNA expression. At time 0, it's 0.3. We measured samples at 12 hours that was not detectable. Okay, so samples were measured there but not detected, and then at 108 hours, there's basically a 7-fold increase in the 24-25. This is concentration in serum, it's not flux. Okay, so I'm not trying to claim that, but it's consistent now with what we're seeing with the mRNA expression. The 125-hydroxy in response to an increase in dietary phosphorus, 244 picograms per mil at time 0, slight drop by 12 hours. By 5 days, there's a 5-fold decrease in the active 125 vitamin D that again is consistent with what the body's trying to do to maintain that homeostatic levels. Other data in the literature, in the animal sciences literature, responses of vitamin D and activation of that, you could say that vitamin D activity is suppressed by excess calcium. This is the shift to calcium. But the animals do respond to that. I'm still kind of curious as to how we created the vitamin D deficiency in our sow herd that created this kyphotic condition. I'm not sure. I have some reasons for that. We can talk about that later. But there's certainly literature basis that showed that the animals would respond much like humans do, or pigs would respond much like humans do to calcium, phosphorus, vitamin D regulation. So kind of a summary that I'll try to stop now on this slide, try to take some questions. Maybe if you work with the FGF23, this is consistent with some of my colleagues in the Madison campus, Wes Pike and Mark Meyer, that are working in this area. They've done studies that actually looked at their mouse studies and kind of modeled the pig study after some of what they've done with the mouse. But what we now know about FGF23, and this is Mariola's slide here, high phosphorus levels would increase in the activation. I think some data that we've shown for that. In the bone, regulation of FGF23, we know that it has to be excreted in the intact form to act on the kidney to alter then the sodium phosphate transporters and the vitamin D activation. One of the reasons, and maybe some in the audience, the reason I'm mentioning this, I suspect this may contribute to why we're not able in the pig to measure the FGF23 in the intact form, is because we now realize that it's glycosylated right at the site of the cleavage that would occur in the molecule. And I suspect that the pig, we have not stripped off that glycylation area and allowed somehow allow the antibody to react with that. So we're still pursuing that. I'm going to kind of stop, I think, with that slide and try to have time for any questions rather than me try to walk you through the conclusions. So questions?
Hi, Dr. Crenshaw. Thank you for a fascinating talk. I'm also pleased that you brought up the fact that when you augment phosphorus and increase phosphorus balance, we think that it goes to bone. But in fact, in humans, a higher serum phosphorus in the Framingham offspring study is associated with increased mortality. And there's also biologic plausibility that a higher phosphorus induces vascular smooth muscle cells to become basically bone cells. So what you're doing is not building bone, you're building vascular calcification.
It's going to ask you real quick, in bone tissue or in non-bone
in
tissue?
non-bone tissue.
Okay.
So I was going to bring this up to Dr. Koh before, but couldn't.
Yeah.
So I'm bringing it up to you. Augmenting phosphorus has to be done carefully and with great study, certainly in humans, because we know in patients with chronic kidney disease, excess phosphorus is associated with incredibly high mortality.
So just a quick comment. Thanks for those comments. I really appreciate that. It gives me certainly another area to think about. So where would those cells in the phosphate accumulation, what tissues in the body would those be more prevalent to occur?
So if you take vascular smooth muscle cells,
Okay.
expose them to high serum phosphorus, you get upregulation of PIT1, which moves phosphorus into the cell, and then that stimulates CBFA1, also called ROCK2, which causes that cell to become osteogenic cell that secretes everything that a bone cell does. So again, just a little caution. Augment phosphorus all you want in pigs, but in humans, I'd be a little careful.
Yeah, well, no, actually the pig world, we're really trying to minimize that for the environmental pollution concerns, and it's very expensive in the pig diet, so we try to minimize it. We still overfeed to prevent deficiencies, so it's still overfed, but not at the level that it's overfed in the human diet. So if you look at human diets as a nutritionist, I think there's a lot of excess phosphorus in the human diet, a lot more than there is calcium, and that was one of the points now that we have to think differently when we switch from animals to humans because of the diets and the way we feed our animals, and we can control that. And then what we consume as humans, we have a lot of excess phosphorus in the human diets. So, thanks.
Follow up, great talk. Follow up on David's question or comment. So direct phosphotoxicity in humans has not really been proven, but the data is pretty damn suggestive. It's not just deposition of phosphate calcification, it's the flux. A lot of phosphate comes in, a lot goes out, you think it's zero balance. It's like sodium. Maybe it's the flux that matters rather than the accumulation. That's very difficult for scientists to get their hands on. So this is what I add to that question. I have maybe a comment. FGF23 mRNA in the kidney. Which cell type is it?
I don't know.
And this is.
It's surprising, it's a surprise we've discussed that the FGF23 went up in that last time period. And one of the explanations, it might be an inflammatory type response. But I don't know if that would go back. Are we doing some damage to the kidneys causing that with what we've done with the diets?
Exactly. You have to flush out all the leukocytes.
Right.
When people find FGF23 mRNA in the kidney, it's in the white cells.
Okay.
The kidney tubule is not participating. And just a final comment, a lot of people have trouble making antibody to pig proteins. I don't know why. We have over 30 anti-clotho synthetic antibodies. We can measure clotho in every species, except the pig. So pig has no clotho as far as the assay is concerned, so you may want to make your own.
Well, we have